reforms it, and then it forms the transition base. So it already forms the transition base that keeps everything from getting there, controls everything that enters into the cilium. And then it is docked to the cell membrane. And then it reforms and regains its function. So when you look at nephronephrosis, there apparently are about 500 proteins associated with the cilium. And there's been diseases, ciliopathies discovered for approximately, I think, a little bit more than 200 of them. So that's expanding. As far as nephronephrosis, there are 20 of them. And what's interesting is they tend to localize to certain places in the cilium. And that is how they affect function. So the most common is NPHP1. And so 20 to 25% of nephronephrosis. And when you look at what causes the trouble in NPHP1, most of it is to deletion or duplication. In this case, deletion. And only 2 to 3% for single base pair changes or smaller changes. And when you look at why, is that during evolution, we have reduplicated multiple areas in this particular gene. And so there are 2 regions that are virtually identical. And then they flank the NPHP1 gene so that they'll form a hairpin and you delete out the NPHP1 gene. So as far as molecular pathogenesis, interesting that it's something that you normally have, but it's prone to undergo this change. So utility of diagnostic test in this case is you establish a genetic diagnosis. End-stage renal disease before age 20 is the predicted information. Renal limited disease in 80 to 90%. Currently no effective treatment. The disease will not recur in the transplant, and oftentimes, in this case, genetic counselor testing other family members and family planning would be suggested. And as with many genetic things, once you identify something genetic, you can find a surrogate marker for it. That can be morphology, that can be an immunostain. So Chris Larson in the lab and Steve Ponset initiated a study on 45 renal biopsies from young patients with chronic kidney disease of unknown etiology that we had tested in the process of validating our NPHP1 assay. And they wondered, were there morphologic changes that were more common in the NPHP1 cases than others? And what they found were there's many chronic changes, but there seemed to be more tubular florets, macula densa-like lesions, diverticulae, TAM-Horsfall protein aggregates, periglomerular fibrosis, and absence of arteriosclerosis. So again, just because you find something genetically, you may not have to keep doing that test. You can find a surrogate marker. So these are ciliopathies, multiple genes identified, NPHP1, most common genetic etiology. The last one is to kind of show a bit of an interesting problem. 22-year-old Hispanic male with hematuria, proteinuria, hypertension, acute renal failure was found to have FSGS. And a sequencing panel for FSGS was requested. This is what our FSGS panel includes in it. And what we found was actually he had 2 variants in COL4A4 on chromosome 2, and he had a single variant on COL4A5 at the same time. So he's homozygous for the COL4A4 and he's hemizygous for the COL4A5. Both of these are causes for Alport syndrome. The trouble with that is it looks like we have a smoking gun. We found some mutations that we think can cause this, but both are rare. They've actually neither been reported in association with Alport syndrome or FSGS. And then there are no previous reports. If I go to EXAC, so some databases that record a lot of variants, they're not reported in those databases. And if I put it through in silico analysis, where you take a computer to kind of predict how it's going to behave, they are damaging and probably damaging. So it looks like it's something bad. There's not enough evidence to call this. And so basically the same for both variants. And so in the end, it was the report I signed as 2 variants of uncertain significance, which is really, really frustrating. And so, but with, we're learning more. And so as more data comes in, we'll be able to do this more. That's why there's a lot of push to kind of aggregate all this genetic information so we know what we're dealing with, because there's a ton of information. And so now you actually have countries that are buying their own sequencing capabilities and actually testing their people. And it makes sense because no two countries are going to be the same, no two individuals are the same. You have to go through a lot, you have to accumulate a lot of evidence to assign whether something is benign or pathogenic. And so this is just a quick summary. It's a lot, very involved. So, as far as sequencing, I wanted to run you through just quickly. The cost is really, really coming down. And so when Sanger, which you probably all have heard, Sanger sequencing, when we switched, Sanger sequencing was basically following what's called Moore's Law, that as we develop more computational power, the cost of that computational power will continue to go down. And when we look at genetic sequencing, so sequencing 1 million nucleotides or 1 million base pairs, the cost is actually dropping at a logarithmic scale very quickly. And so the cost of and capability is coming down. And that's a lot has to do with generations of sequencing that we've been able to do. So there's the second generation, which is mostly short reads, and third generation, which are mostly long read. And to kind of highlight the amount of data that you're generating, so you a typical human genome, if you just take one copy of that, it's 3 billion base pairs, approximately. And if you're going to do just look at exons only, you're really only looking at 1 to 2% of the human genome. So it's really not as much information as you think it's going to be. As far as when you look at the amount of progress we made, so the Human Genome Project, 13 years, 23 labs, 500 million, 3 million bases. And now we have platforms that can do 48 human genomes in 45 hours, one machine, at about $1,000 a piece. And so when you look at the data scale, one genome at 30x depth is about a terabyte of data. So you're looking at just huge infrastructure in order to be able to manage the data as well. And no two people are put together the same. So this is 1,000 Genomes Project data. The typical human genome differs from the reference genome by 4.1 to 5.0 million sites. So what's normal? There's no normal. We'll all have normal. And so most of those are taken up by single base pair changes and short indels, but when you think about structural variants, deletions, and copy numbers and inversions, they take up more base pairs. Mostly what we do in the lab is DNA resequencing, in the sense that we're sequencing somebody's DNA and then we're trying to align it to a reference to make sense of it. You can do de novo sequencing, and as you've heard from Dr. Barsoni, also RNA sequencing or transcriptome sequencing, and you can do that on a single cell scale, which is pretty amazing. Also, there are limitations to the technology. So you're using a polymerase to essentially sequentially read through DNA. And so if it's GC rich, it's really hard for the polymerase to make it through there. The other part about it is that when you're looking at segments of DNA, and you have to bioinformatically determine where it came from, there's a lot of sequence homology in the genome. So basically, about 50% of your genome is repetitive. And so when you're only looking at small pieces, it's hard to map that. So with Sanger sequencing, you basically use base pair, dideoxy base pairs that constipate the polymerase and it'll truncate it. So you generate different size fragments when you're doing that, and you run those out onto a gel and you can see what the sequence is. We now use actually cyclosequencing, in which you can do that all in one tube, and you have fluorescently labeled probes. And that's what an electropharogram would kind of look like. And if you get a mutation in it, you would see something like that. And we sequence forward and reverse so that you can check yourself. And if you get a deletion, it changes the frame. And that's kind of what it looks like when you get a deletion. And if you were to slide it over to where it was, you can kind of see where it matches up. So now when we start to look at other generations of sequencers, when you look at Sanger sequencing, you're actually looking at a population of DNA molecules. When you get into the next generations of sequencing, you're actually looking at each individual individually, and that's one of the things to kind of take home. Next generation sequencing has two components for validation. There's a wet bench, which are actually just what are you doing on the platform, what are you doing in the lab, and then there's a dry bench bioinformatics platform, and then there's a part in which it's report generation and entering variants into databases. All of these components have to be validated. This is an example of a short read technology. I wanted to briefly show it. So you fragment the DNA, so this is chromosomal, you fragment it, then you add on these barcodes, so you put something on here that enables you to identify that sequence later. And then you can amplify this and sequence it. So you copy it again, and now you have a forward and reverse strand on there. And so just like Sanger, you can forward sequence and reverse sequence it and confirm it. You can imagine that one molecule you're not going to be able to see with the camera, so you want to do this clonally, so you make a big spot on them so the camera can pick it up. And you do millions of them on the same flow cell. And so when you sequence this, each time a base pair is incorporated, the camera picks it up. And again, you do that over, you do forward, you do reverse, and you do it over the entire chip. And then the computer then uses an algorithm to separate out these. And then you also map these. That's one example of a sequencing platform that does short read. Probably the most common platform used. This is a different one that's starting to do some really interesting things. This is Nanocore. And what you do is you have a lipid bilayer and you have a channel in it, and you thread a DNA molecule through that channel. And as the DNA goes through it, it changes the conductance, and you can tell where the base pair is. And this one's actually really cool. They have a sequence there that's the size of a USB chip, or a USB drive. And you can actually take it out in the field and use it, and it plugs into your computer. And they're using it for looking at epidemics. And so it's pretty awesome. This is also a single molecule, and the reads are huge. They get up to 50 kb. And so really, really big. This is a different one, of Acidic Bio. And they actually take a DNA polymerase, put it at the bottom of a well, and run the DNA through that. And as the molecule, as the base pairs are incorporated, it is phosphorescence, and you can detect the sequence that way. In the talk, I put the links, so if you want to see more about these platforms, you can look them up. If you look at the data on these, usually what you're looking at, this is ApoL1, and here is actually the exon, the intron, exon, intron, and you can see that in our assay, we actually analyze just the exons. And what you're seeing, these are the individual reads, and you're seeing that we've covered that entire exon 100%, and we've read it to a pretty significant depth. That means you sequence the same area multiple times. So again, you look at individual molecules as compared to Sanger. So if I look close, this is a read, that's a read, that's a read, that's a read, and you can see that for all the ones that are here, this is homozygous, and I read it 198 times at this base pair. Same thing, here's one that's a heterozygous. I read this particular base pair 175 times, and it's heterozygous. So, and when we talk about sequencing costs, it's a big deal that it got below $1,000 for sequencing, but the truth of the matter is that it doesn't include bioinformatics, interpretation, and confirmation. And you know, some people say that it's $1,000 sequencing, but it's like a $10,000 bioinformatic problem. And so, also, many times, we're still trying to figure these things out, and so we'll sequence family members or do other things as well. So, utility of genetic testing in this case, genetic diagnosis, currently no effective treatment, the disease will not recur, genetic counselor consultation, testing with other families, and family planning. We actually did test this patient's mother, and she had the col4A5 heterozygous, and she had col4A4 heterozygous. And so he's got a double whammy potentially. And so she didn't have disease, but it gets to be kind of interesting. As Dr. Barbasoni said, there's a tendency for molecular biologists to say that the answer is in DNA. It's DNA, it's DNA, it's DNA sequence. But the truth of the matter is it's a lot more than that. It's the DNA, then it's also epigenetics, and then it's also, okay, how was the protein traffic? Did the protein traffic the way it was supposed to? Huge, all over the place. So in the end, it is data integration. And so the last thing I wanted to talk about, just because it's interesting, basically the summary is that it's rapid progress in molecular genetics and bioinformatics. I think this will continue to go. We're getting better at handling the data, but we've got a long ways to go. Some issues with patient access as far as cost of testing and reimbursement is still a big issue. We do most of our genetic sequencing eat the cost, and then this is an interesting one that I wanted to briefly touch, was circulating cell-free DNA, which you guys have probably already heard. So you can imagine when in your blood you have cellular DNA, you also have extracellular membrane vesicles that bleb off from the cells, and you have protein-bound and free DNA and RNA. And you can use this for testing, non-invasively. And so you can do testing on a fetus from circulating fetal DNA within the mom's blood. And so that's one example. And that's what's interesting about cell-free DNA is your circulatory system is sampling lots. And so if you can leverage circulating nucleic acids as a diagnostic tool, that's pretty amazing. So they're already using it for cancer. There's actually now a company that will, if you get a transplant, renal transplant, you can tell circulating free DNA that arose from the donor versus the recipient. And you can detect rejection much earlier than you could on biopsy. And so, again, there's some really neat things happening and a lot of opportunities. And that's it. And thank you very much.
So thank you, John. Yes, Helen.
First, I would like to make a clarification about John Dean and then a neuropathologist doing nephro work.
I did say that. I said he's working in the nephrology or nephropathology field now.
No, there is another section in Arcana, and he's heading neuropathology. But he is among the very few people who has a double certificate. One is in neuropathology and the other is molecular pathology. And he runs a CLIA-approved lab for diagnosis of kidney disease. So I know John, he's very modest. He never talks about his publications, so I just wanted to make that. So John, this was a great talk. Can you give us an estimate about how many biopsies Arcana does now and how many of them are being tested in the lab for genetics?
So currently, I think Arcana will be up around 15,000 or so per year on renal biopsies. So it's a huge volume and a really neat opportunity in that sense as well. And so there's lots of studies going on. The interesting part about the sequencing is I would say that as far as sequencing goes, we test, I would say, much less than 1%, 0.5%. And part of that is actually patients are interested in it, but insurance companies in the US view it as experimental. And that they are not willing to pay for it, will not pay for it. And so it's an interesting problem. The reimbursement is based on individual genes, like you would do in Sanger. If you were gonna, for Alport's test col4A5, you can get paid for col4A5 sequencing it. If you do a next-gen sequencing assay that's way less expensive, you can do 50 genes for much cheaper. They will not pay for it.
So these are the realities, but John is an example of how our job as a nephrologist is changing, and we cannot cover all bases. So the renal pathologists have to work together with other people like bioinformatics or geneticists, and either in the same lab or adjacent labs. And we need to understand the technology and how to interpret it, which is the biggest challenge. So one of the challenges is those uncertain, significant results. You send something for genetic analysis and you get back uncertain results. Yet the patient is very sick. We have seen the biopsy. That's the value of the renal biopsy. But the renal biopsy is not uncertain. If somebody has 50% of interstitial fibrosis, the kidney is sick. And if you have an Alport diagnosis, the patient has Alport, even though we never test for the specific mutation, because in Alport, in particular, they are so individual, the basis, that there are thousands of patients tested and there is not a hotspot, right? So then there is the other complication. Those of you who come tomorrow to my talk, I'm talking about congenital nephrotic syndrome, where you don't only have one mutation, but you have 2 or 3 collaborating. Sometimes it's the collagen 4 gene, and it collaborates with Neil-Patel syndrome mutations. So we are learning that the prognosis in different patients who have also phenotypic variability is not only one gene mutation. And you're saying, what's normal? We have curly hair, it's a mutation. We have blue eyes, it's a mutation. We are full of mutations. Not all of those are bad. So we are learning a lot, even as we speak, and we understand from these not necessarily definitive diagnoses, we understand why the renal biopsy may show what it shows.
Okay, yes, you want another comment or question? So I have a short, I would like to have your opinion on this, because in Europe, things seem to be quite different from the way we can approach mutations, because in pathology you can look at somatic mutations, but you're not allowed to go into germline mutations. So from the data you presented, we could probably do the FISH, and then on the other hand, I would ask myself, it would be really necessary to establish the FISH if you have maybe one case every year where you could apply it, and then it would only be 20%, so you'd have a positive FISH result every 5 or 6 years. And I don't think our molecular path lab would say, well, yeah, we can certainly do it, it's a great thing, because it's very rare, but all the other things are in the hands of the geneticists. So how is it in the US?
I think I would agree. I mean, I think anytime you're bringing up an assay, it's that how many samples do you get versus what's the cost, multiple costs to maintain that. When you're doing more proficiency testing samples, then you're actually doing true assays, and then it's not. So it's always a balance. In the United States, I think the thing that's happening is that there's a movement. Essentially, one of the contributors, I think, is trying to centralize a lot of this. So there are big sequencing centers that can do this in a very cost-effective manner. And so it gets to be an interesting problem, is you just go ahead and send your material in, and you can get the genomic data back, but you don't have that capability in your lab. And so, again, interesting to kind of navigate.
Yeah, but in your case, you can order the test, because in Switzerland, I mean, we would have to inform the clinician then would have to contact the geneticist or leading geneticist, and they would have to order.
But that's the same, that's the same.
Yes.
I mean,
Yeah.
The clinician.
right, the clinicians order the test, the lab performs the test. Yeah, yeah,
It's the same.
yeah,
Okay.
yeah.
Yeah, you have to get genetic consent in order to be able to.
I mean, what you mean is that the pathologist may discuss and suggest to the clinician that in this particular setting, such and such molecular testing would be, of interest. But then ultimately, in terms of placing an order, at least at our institution, it is up to the clinician.
Okay, so any other questions? So I think we had a great session, great first session. Unfortunately, we're 30 minutes late. So we'll have 20-minute coffee break, and then we'll, short.
There's coffee downstairs, and instead of making half an hour, we're going to make 20 minutes of a break. I hope it's time enough for everyone to go to have a coffee in the